I started at Purdue in 2006, and at that point in time, the states to the west of us had revised their nitrogen rate recommendations. They had found that the yield goal-based method that we were all using at the time wasn't a real good way to make nitrogen rate recommendations. In fact, there was no relationship between yield achieved and how much fertilizer nitrogen had to be added. And so their new recommendations were based on hundreds of field trials. And in Indiana, we just had a handful of nitrogen rate response trials that had been done since 2000. And so when I came, the corn specialist, Bob Nielsen, and I got together, and we talked about how we would build a database to make nitrogen rate recommendations. And one of the first things Bob Nielsen said to me was, I'm too old to drag a plot planner and a plot combine around the state and do lots of nitrogen trials. And he said, and as a matter of fact, I'm too lazy to do that as well. So he said, but I've been doing these large-scale strip trials, and the results are surprisingly good, and I'd be in for doing that on our Purdue farms and farmer fields. And so we started doing nitrogen rate experiments. Probably about 2/3 of them have been done with farmers. All of them are field scale or large scale, 10 to sometimes a couple hundred acres. Most of them are in the 30 to 50 acre size. Anyway, so I'm going to talk a little bit about how we did that and some of the other studies that we've been doing with plant population and some other things. My presentation will be posted on this soilfertility.info website, and I understand you'll be able to painfully rewatch the video of this if you are compelled. But there's some websites in here that I provide the URL, and you might want to pull it up just to get that URL instead of trying to scramble to write them down. Our goal is to provide unbiased answers to practical crop production questions under real world conditions. Purdue has 7 good agronomic farms scattered throughout the state. Ohio State has several. There's a few university research farms, but they're not really enough to represent the breadth of soil and environmental conditions and management systems that people use. And if you look here in Indiana in particular, there's a lot of crop production in this central area, and there's really no farms there that represent the soil types in that region. So the only way we can get data from those areas is to collaborate with farmers and work on their fields. The benefits that we found, again, it expanded our ability to answer these practical questions. We found that the variability in these large strip trials was actually 3 to 5 times less than what we encountered in traditional small plot university research. So going to the large scale had a lot of advantage in being able to detect small differences and to better represent the responses we saw. And that's really was unexpected. And in fact, some of our colleagues don't believe that there's that much of a difference, that they're that much better in reducing the variability. And I think it has to do with the fact that if you place a trial in a field properly, you kind of average over all the variability. Whereas when you have a 30 by 30 plot, a lot of times that can be in a particular soil type or topography or something where it's just a lot different than the other replications of that same treatment. I think the farmers that we work with and the CCAs and the industry people have more confidence in the recommendations when it's done at the field scale with traditional equipment the farmer's equipment or our Purdue Ag Farms larger scale equipment as well. And just working with farmers, their consultants, some of the educators, increases our collaboration and cooperation. And those folks work with their neighbors sometimes and spread what they've learned on their farm. And so it's overall, it's a better working relationship. One of the things that's facilitated this is the technology. And when Nielsen said he was lazy, he was remembering the days that we spent out in the field with flags and tape measures and wheels, measuring off things and dumping out planter boxes and cleaning out the fertilizer tanks, because it was hard to do field scale research before we had technology. But now with GPS, RTK, aerial and satellite imagery, digital soil maps, and we can get things done a lot quicker and a lot easier and a lot better. Here we have a field that's planted on the contour. We did a trial in, and I can't imagine how we would have done this any other way other than by using the RTK to put these plots in. You see the different colors represent different replications, and the strips within each color ended up being the treatments. This was also a poultry litter experiment, and we can do with RTK, you can put out pre-plant treatments and fall application of nitrogen and things like that really easily, and not have to go out there and mark where every treatment is, and then pull the flags up and plant and then put the flags back, etc. Yield monitor, of course, was really helpful as well. We try to calibrate all our yield monitors because, there can be a lot of error in a yield monitor that's not calibrated. And for research purposes, we definitely encourage the grower to calibrate with us the yield monitor at the beginning of harvesting a trial. That's one of the impositions that we put on the farmer. If you have a prescription map or and the harvesting process itself really is no different than what a farmer would do normally. But to calibrate the yield monitor to the level that we like it to be calibrated for research does take 45 minutes to an hour at the beginning of the day. So that is one thing that's a little bit different than their standard operation. Nowadays, most of the trials that we get, we interact with the farmer or their advisor, and they send us the locations of the potential fields that will be used for a trial electronically. And we look at the aerial imagery, the NRCS soil maps, and we lay the experiment in there and see how it'll fit and if it's going to be suitable or not. Some fields, by the nature of their variability or their size, are not necessarily suitable. And you see this sandy soil in northern Indiana had a couple different soil series here, and then the majority of the field was a Maumee sand, and we placed one replication entirely within these two different soil types so that we blocked out that variation, and then the rest of the reps are in this Maumee sand, and we clipped out the middle area where water was draining to that woodsy area. And with RTK, we can go in and plant these plots with a prescription map, and the farmer just moves from one side of the field, the north to the south, and everything's planted. Then the treatments are applied later in the year. But being able to look at the field from the aerial perspective with the soil map is really helpful in determining how to utilize that field or which field is best for the type of experiment we're trying to do. Yield data is important, and GIS allows us to use irregular shaped fields. In the past we always looked for rectangles, If you go to our Purdue farms, they cut up these big fields into 10 to 15 acre rectangles to put their plots in, and everybody's always looking for something that's square. By using GIS and both the application of treatments and the harvest of yield, you don't have to have irregularly shaped fields. If you have a big wet spot, if the raccoons get into a particular area of the field, or deer, or somebody drives a Jeep out there to drink beer, you can remove that variability that has nothing to do with the treatments from the yield file and kind of clean up your experiment. And then what we've just started working on with basically about 300 of these trials that we've conducted in the last 11 years is taking a look at these nitrogen responses and plant population responses by soil type. So you have this big spatial data set. You can look at a particular soil type, say here, and look at the response to the N rate. What's the optimum N rate in that soil type versus the other soil type? So these larger scale trials give you the opportunity to look at approaches to variable rate nitrogen and seeding rate. We try to keep it simple. Most of our experiments are 5 or 6 treatments, different starter fertilizer, or different rates of nitrogen, or different seeding rates. But it can be fairly complicated. This was a couple 100-acre trial where we had 5 seeding rates, and within each seeding rate we had 5 nitrogen rates. So the question was, if you use a higher seeding rate, do you need more nitrogen to maximize yield? And in this study and several others like this, we found that the optimum N rate was consistent across seeding rates, so it didn't increase with more plants that we put out. So it can be pretty involved, but most of them are fairly simple, where we only do 5 N rates or only do 5 seeding rates. I mentioned earlier the technology. Almost everything we do is with a prescription, and so it's seamless for the operator. We use GPS to tell the tractor where it is and when to apply 100 pounds of nitrogen and when to apply 200 pounds of nitrogen. And with variety trials or starter fertilizer treatments or biological enhancements, using RTK to go to the particular plots makes it easy, so you don't have to clean out the boxes or clean out the tanks. We try to get an as-applied file as well. Some of those actually measure, say, flow of a fertilizer material or the number of seed dropped. Others measure shaft speed. But in any case, we try to get some confirmation of what we've done, because occasionally errors occur. And with that as-applied file, you can catch those errors. All right. So for us in our research, we wanted to do multi-year, multi-location trials in sufficient quantity to be confident that we have a reliable answer. So these aren't one-offs. We do some one-offs where somebody says, I'm interested in this, and we'll help them set up a trial, and we'll do that 2 or 3 times maybe. But we're really focused on doing a lot of a certain kind of trial so that we could build a database to make recommendations for everybody. Each of these trials has to be replicated and randomized, which means you have to have more than one of each treatment, so we can't just do a side-by-side. And randomization means we're not going to have like a systematic arrangement of the treatments. Sometimes there's systematic variation in the field, and if you line your treatments up a certain way, you'll favor one treatment or another. And think about tile drainage, and if you get a particular treatment that's always over the tile drain versus the other treatments that are between tile drains, that's going to influence the effects. So randomization gives every treatment the opportunity to be in a particular area of the field where there may be some systematic differences. And that allows us to do proper statistical analysis. So we have some sort of confidence level in the answers that we're providing. You can do pretty simple experiments, yes or no questions, starter fertilizer, different types of starter fertilizer, foliar fungicide, cover crop, seed treatments, things where it's more or less a yes or no, or one treatment versus three other different treatments. And then you have rate experiments where you're looking to identify what's the optimum nitrogen rate or optimum seeding rate, or could be the optimum rate of fungicides, any kind of a thing where you have multiple rates of a different treatment. Bob Nielsen has an on-farm research webpage where it explains kind of the requirements of these trials. He has a bunch of example research protocols. He has some links to the summaries that we've developed from the research as well, and contact information for the different specialists at Purdue that do on-farm research in different topic areas if you're interested. Here's the different protocols. So fungicide, soybean seeding rate, row spacing, corn seeding rate, corn end rate, soybean fungicide trials. Just ones that we have established protocols for. So I'd like to talk about the nitrogen rate experiments first, because that was kind of our first endeavor. Here's a farmer cooperator that was flying over some of his experiments or land, and he got a picture of our N-rate trial here from the plane. In this particular trial, we had starter only, 29 pounds of nitrogen per acre, and then we had 50-pound increments up to 229 pounds of nitrogen total. And you see the 5 treatments here. You can obviously see the 2 low treatments because the corn is very deficient in nitrogen. And then there's 3 replications. So here's the second starter only, and the third starter only, and the second 79, and the third 79. And the higher N rates are not showing much difference there. So most of the trials, the end trials, are going to look something like that. Most of the time when we work with farmers, they're not interested in doing starter only because they're going to lose lots of money. This particular trial, the farmer was interested in doing the low rate, probably lost about $5,500 out of that one field due to the reduced yield. In order to do these trials, in order to determine the optimum rate, you got to have something that's too low, and you got to have something that's too high. And so we try to gauge the high and the low so that the farmer's not losing too much money unless they're just interested in seeing what those starter-only treatments would look like. We need to have at least 4 rates. We like to have 5. You need at least 3 reps. It's nice to have 4 or more. It just depends on how many treatments there are and how big the field is and what the size of the equipment is. And as I said, you gotta reduce yield in this type of a trial. Now, if you're doing a yes or no, then it doesn't necessarily have to be any different. You're not necessarily picking treatments that you know are going to be negative. You're usually using the standard treatment as your comparison. But for the rate experiments, you need to go below what they normally will utilize, and hopefully a little bit below what's optimum. Equipment compatibility is important because most of the time the treatments that are imposed have effects on their neighbors. So you need to harvest out the middles of these plot areas, and you want to be those middles and the in-betweens to be full header width. You don't want a guy with a 12-row combine picking 8 rows in a 100-acre field. You want to be able to take out the middle 12 and then the next 12 and then the middle 12 and the next 12. So we ask them about their planter, their applicator, their combine, and we try to find an arrangement that allows us and them to work efficiently. So you see here with a 12-row planter, a 12-row applicator, and an 8-row combine. In order to be efficient, we need to have a 24-row plot, and that's where you have to have not too many treatments and not too many reps when each plot is 24 rows wide. So here's the earlier example, 16-row planter, 8-row applicator, 8-row combine. Just start in the middle of a plot and harvest the rest of the field. Applications, these 16-row plots, entire run of the applicator, so not a problem. All of our cooperators get their individual trial results. And here we have all the details that we gathered from the farmer, what we did, and then we draw out and represent the response. So this is an N-rate trial. We started at just shy of 100 pounds per acre. We went up to about 2 1/4, and somewhere here at about 180 pounds of N was the optimum N rate. So we provide that to the farmer. We utilize the data we collect on their farm in a big database that's anonymized, and we don't share the specifics of where this farm was and who the cooperator was or anything like that with anybody. So they aren't really singled out, and they contribute to the overall database by allowing us to use these response curves. Most of the people that we've worked with can work with us many years. This person started in our first year, 2006. We didn't have a trial last year, but we had trials every other year up till 2015. If you were in my manure talk yesterday, I showed an example where we were looking at nitrogen response on fields that received dairy manure in the fall of the year. Before we did that, he wanted to know what the response was in a corn-soybean rotation on ground that didn't receive any manure for 2 or 3 years prior to conducting the trial. So we did nitrogen response experiments in a couple fields in 2006, and you see the response there pretty consistent. 2007 we lost our trial because it was too wet in that particular field, but in 2008 we conducted it again, and we were able to split out the 2 soil types in this field, the cyclone and the salina, and look at the response in both of those soil types, and it was pretty similar to 2006. And then in 2009 in another field we were able to do the same thing, and you see the responses there. And he had pretty consistent results. He felt pretty good that he had defined the optimum N rate in his particular system on these soils that were very similar across his farm. But we encouraged him to think about what some other farmers had found in similar soils, and you see that their responses and his responses fit in all together. So these were pretty consistent results. We don't always find that, but he was pretty comfortable in that the optimum on his farm was about 200 pounds of nitrogen per acre, and the other farmers that we included in the database, in addition to these 2 that were near him, the optimum N rate was on average about 200 pounds per acre as well. So he felt comfortable with our general recommendations. He contributed data to them. He found his farm was similar to what we considered representative for those areas. And then we moved on to looking at nitrogen response in his manured fields, which turned out to be a lot more variable than this particular response. So all of these experiments are summarized in these nitrogen management guidelines for corn in Indiana, and this is the URL for this particular publication, and you can get that from the soilfertility.info website. In Indiana, we were able to do enough trials to separate our responses by region of the state, where we have some big differences in soil type and a little bit difference in rainfall from north to south. Here are the recommendations in the southern and western part of the state. The optimum N rate with current prices is around 180 pounds of nitrogen or less. Over here in northeast and east central Indiana, it's up around 220 pounds of nitrogen per acre. And we were only able to do this by conducting a lot of trials in each of these areas and determining that their nitrogen responses were different. If we just relied on our 7 Purdue farms, we wouldn't have any recommendations in southern Indiana, in north central Indiana, or in central Indiana. And so only by using results from cooperators in these other parts of the state were we able to build enough of a database to make those recommendations. All right. The economic N rates, of course, will vary by grain price and fertilizer costs. So in that handout, we have charts that show you what the optimum rate will be for different combinations. So 350 corn and 50 cent nitrogen, it's about 212. If corn were to increase to $5.50, the optimum N rate for corn after soybeans in northeast Indiana would increase to 230 pounds of nitrogen per acre. So building these response curves allow you to look at the economics and determine where you want to be on that yield response curve to make the most money. Some people are interested in these interactive tools, and Iowa State hosts a corn nitrogen rate calculator that's based on the data from the individual states. So you see Indiana and Ohio contributes to this database as well. And so you can look at the responses, the average responses based on different corn prices and nitrogen fertilizer prices here, and then see what the optimum N rate would be here. And in this particular example, with 350 corn and 80 cent nitrogen, it's as low as about 150 pounds of nitrogen per acre, to drop the price of N to 20 cents, and it's as much as $200 per acre. So this tool allows you to kind of change those prices and play some scenarios out and get that optimum nitrogen rate. We focused on nitrogen, which typically is the second most expensive input in a corn budget. The top most expensive input in a variable rate budget is seed. And Bob, of course, is an agronomist, and he was very interested in looking at yield response to plant population. So we took the same approach that we used with nitrogen rate trials, and we started, I think, in maybe 2009 and 10 in earnest. And we have it says 81 field scale trials looking at optimum seeding rate and plant population response. These results are summarized in that publication I just showed you. Here, under normal conditions, 67 field scale trials resulting in an average population for maximum yield of a little over 31,000 plants per acre. I noticed Emerson yesterday was talking about some of the the 50 trials they did in Illinois, and it was 33,000 in his particular experiments. Again, the interest is really looking at this in economic terms, and Bob projected a number of seed costs here with grain prices, and adjusted that optimum based on the economics, producing the most profit, not the highest yields. In this handout, you'll also see a number of trials that we did where we compared hybrids that the seed dealers said would be more responsive to higher seeding rates than other hybrids. And we had some other experiments with multiple hybrids that were supposed to be more responsive than others. And basically we found there was no relationship between the population needed to maximize profit or yield and what the seed dealer told us about the particular hybrid that they were selling, and it being more or less responsive to the different populations. In some of these trials, we try to collect additional information, pre-side dress nitrogen test, Illinois soil nitrogen test. We do some tissue testing sometimes, chlorophyll meter readings, end of season stalk nitrate test. If you were in the manure talk I did yesterday, we had a very big database with an end of season stalk nitrate test that was built from these large-scale strip trials that we did on our Purdue farms and with farmer cooperators. A lot of times the advisor to the farmer will be interested in looking at PSNT or stalk nitrate relationships to the yield response, and they'll actually collect those samples out of each of the treatments and help us to build a database to evaluate those as ways for making recommendations. So some other topics that we're transitioning to. I think we feel comfortable and confident in our nitrogen rate recommendations, and we're going to kind of not do too many of those in the near future. Same with plant population. We've started to look at 2x2 and pop-up fertilizers, trying to build a database there. I think we only have 14 or 15 of those trials. Late season and application timing and placement, so Y drops versus dribble and splits between V4, V7, V12, things like that. This year we're going to start a series of sulfur experiments where we look at nitrogen with and without sulfur to try to ascertain how likely sulfur deficiency is to occur and what the potential yield benefits are of including sulfur in the nitrogen fertilizer. And then we have been looking at some in-furrow biologicals as well. There's lots of questions that people have. I think we found the best way to address this is to work with farmer cooperators and in our Purdue farms and do these large-scale trials. You have to be committed to doing the same type of experiment over a number of years at a number of locations to really build a big enough database to make recommendations. But I think in the end it's worth it because you have a confidence in the result, and that result is lasting, versus doing 3 or 4 experiments in a limited geography and then making a conclusion based on that.